Hi everyone, and thank you for watching my presentation. This is going to be on the Leucine Zipper. My name is Brittany Hamlin, and this is for class Chem 330. This slide is just going to tell us a little bit more about the Leucine Zipper because most people wouldn't know what that is. First of all, it's a part of transcription. Transcription is the conversion of DNA to RNA, which is one step in protein synthesis because translation is taking RNA and turning it into a protein. Because leucine is an amino acid, it makes sense that this is part of protein synthesis because ultimately protein synthesis is building of amino acids. Amino acids are the building block of protein. Leucine acts as a hydrophobic core for polypeptide chains that again lead to the formation of protein. Hydrophobic means anti-water, and this hydrophobic core is found toward the innermost of the cell so that water from the outside of the cell cannot interact with it. The leucine zipper is composed of tightly bound alpha helices, and because it is a coiled coil, it makes the leucine zipper very stable, which I'll go more into on the next slide. Some of the examples of bonds are hydrogen bonds and ionic bonds. There are also many more. Here's a little bit more about the alpha helix. Hydrogen bonds are what make the helix very stable, and it's actually the connection of a hydrogen bond between NH and CO. In the top right hand corner you can see a picture of how this happens in a polypeptide chain. The CO group of residue I, this is an example from the book, forms a hydrogen bond with the NH group of I plus 4. So as you can see, the oxygen in monomer number 1 connects 4 down to the hydrogen. These two are attracted to each other and are what really make it be a helix and be a very hard chain to break, which is really important for the building of protein. Because these are two alpha helices bound together, this is referred to as a superhelix or a coiled coil. This example is actually left-handed despite most helices being right-handed. Some examples of helices that make it a little bit easier to relate this topic to are wool or hair, which are also called keratin, and collagen. This makes sense because if you think about our hair and skin, all of these are found in our hair and skin especially is built for protection, and you hear about proteins in our skin and different makeup or skincare products that contain keratin or collagen. These are usually thought of to be the strong suits. So if you think of that keratin and collagen are strong, it's easy to relate them to alpha helices, which are tightly bound and very stable molecules or chains. Part of the assignment was to find a replacement for leucine. So with that being said, of course it must be hydrophobic if it's going to replace leucine. So an example that could replace it theoretically is valine. Valine has a very similar structure in comparison to leucine. Valine contains one less carbon than leucine, as you can see in the pictures to the right. The top one is leucine, and as you can see from the hydrogen in the top left corner, it branches off 1 and 2 times and then in 2 more. Whereas the bottom one only branches off the 1 time and then into 2 more. But because they're so structurally similar, most likely valine would also contribute to hydrophobic tendencies, but also the electrostatic interactions with glutamate and lysine, as you'll see later in the presentation. An example of a destabilizer that would potentially destabilize leucine would be serine, because serine is a charged amino acid and it would destabilize the hydrophobic core. With the OH group on the bottom as the side chain, that's also called a hydroxyl group. The hydroxyl group is the side chain for serine, and these are hydrophilic. So of course, as we keep emphasizing the hydrophobic tendencies of leucine, a hydrophilic side chain would definitely destabilize and would destabilize the core of the leucine. So to talk a little bit about the electrostatic reactions, I wanted to define them first. Because electrostatic reactions occur when one side chain is positive and the other is negative, this is why leucine has the ability to bond with side chains of lysine and glutamate. As you can see in the picture here to the right, leucine is in the middle, as we know, because it's the hydrophobic core, and glutamate and lysine are the two that are on the sides there. Another explanation for this is that lysine is more basic, whereas glutamate, another term for glutamate, has the word acid in it, so we know it's acidic. So because you think of positive and negative reactions and acidic and basic reactions, that's why this works. So a substitution for lysine and glutamate would have to obviously match up with the description that I was just speaking about. Because in order for the leucine zipper to still properly work, the amino acids would have to be ones that work with the electrostatic reaction. So two examples of that are aspartate and histidine. As you can see in the picture on the right, this is the binding of aspartate and histidine. You see the hydrogen, again, bonding with the oxygen with a hydrogen bond. Aspartate in this case is the acidic one, whereas the histidine is basic. And so this again allows for the electrostatic reaction, and also one being positive and one being negative attached to the leucine to remain the same, again, while attached to the leucine molecule. Thank you very much for watching my presentation. I hope you learned a lot.